Good morning, everybody. Thank you very much for coming here. We are going to have the pleasure today to present Raman Quantification in the pharmaceutical industry. What is the challenge of the technology, as well the implementation, regulatory implementation, and some examples on P80. Okay. Raman Spectroscopy is very well known for most of you, certainly, but in case that there is some people that are fresh on the material, I would like to introduce the technology. Raman Spectroscopy is based on light. Basically, it's a technology that produces a fingerprint of the material. It's based on the vibrational states of the material. So what you have is basic information of the material. It's not trace detection, unless that can be used for that is necessary. There is techniques specifically designed for that. But it's, as a general introduction of the technology, is not conceived for trace analysis. Many of the advantage of the technology is that perform the analysis directly through the container. So that is something that is very well received, especially for qualitative analysis, when you want to receive the incoming material and you want to know what is in there. But as well, is very much used today for the capabilities of the technology to go through and understand what is going on the process. So many people are today using the system, the technology for process control, for crystallinity, to understand what is ongoing on the reaction. So very quick summary today, what are the challenges of the technology? The technology in the pharmaceutical market is broadly used today. It's one of the technology that is advances the most in terms of introduction of the technology, even if the technology has been on the market for quite a long time. Today, most of the time, the technology is using on the qualitative analysis. So what you want to do is to identify what the material is because of the characteristic of the fingerprint technology. But more and more, the technology is moving to quantification. Spectroscopy, Raman Spectroscopy is a technology that allows you to have precise quantification modeling. And that is the reason why more and more the technology is used today on the P80 and quality assurance environments. Raman spectroscopy in the pharmaceutical market is extensively used in the warehouses. Products like the NanoRam from B&W Tek is intensively used for the identification of the incoming material. Basically, you receive the parcel, you identify through the packaging what do you have in there, and then you have the system already ready to go into production, almost. It's intensively used in the laboratory environment. Sorry, the laser pointer is not very powerful. It's intensively used in the laboratory, specifically in R&D environments, mostly for characterization. It's extremely helpful as a complement technology for spectroscopy like infrared or near. And is very broadly used in polymorphic identification as well as patent defense. If you have two polymorphic forms in the production chain, you want to be sure which polymorphic form do you have. You want to have technologies like Raman to guarantee that you are in the right process and that you don't have any problems with patent issues, for example. Intensive use in quality assurance, mostly due to the characterization of the material, a lot of use in process control, and more use on the kinetic characterization of the pharmaceutical process. The benefits of handheld technology for identification, purely qualitative analysis, is just that it's really simple instrumentation. It's just a handheld device that can be relatively low footprint. Is introducing a lot of changes into the warehouses. Now you don't need to open the material to double check whether you have in there. Directly the material arrives, you open the first container, the second container is still intact, then you can do the analysis directly. Easy ensemble of operation. You don't need to have a specific qualification to operate it Raman technology. Most of the time, the qualification is just a single day. So it's really straightforward to implement it. Reduce all chemical exposure. If you don't need to open the containers, is quite a clear advantage. If you can identify what is ongoing in your bag or in your reactor, you don't need to have any kind of chemical exposure. Fewer laboratories delay, so you need more and more you need to increment, or you have an incremental speed on what analysis you are doing in the lab. If you can do the analysis on the spot, and you don't need to send the samples to the warehouse or from the warehouse to the laboratories, it's significantly advanced on the production process. Free laboratory time. quite important because laboratories are quite expensive, and reduction of transcription, so less errors, as my colleague was telling before about the potential errors that you commit when you are doing the production chains, okay? That is the advantage of handheld or portable device in general, okay? Specifically, when you are talking about validation, the Pharmacopoeia is quite simple in terms of what do you need to do for validation purpose of this technology. For pure validation of the material, the only thing that is required is the specificity of the technology. When you have something on your warehouse and you want to identify what it is and have an insurance or assurance, complete assurance of what's going on, or what is this material in your house, the Pharmacopoeia is quite explicit. You need to demonstrate it, the specificity of the technology. This means that when you try to validate this technology for purely qualitative analysis, is straightforward to validate it. So the process from purchase, the instrumentation, to implementation is just a matter of few weeks, couple of months, to be able to have the instrumentation on production. A different thing is the quantitative analysis. Quantification is always much more complex, specifically when you are trying sometime in the pharmaceutical environment to replace other technologies. For example, when you are trying to replace an HPLC, that is the sacrosanctum instrumentation for most of the quality assurance laboratories, and then you try to replace that with an easier technology as Raman, then you have a lot of confrontation sometimes with the regulations, because you need to justify in a very extensive way how you are going to do this kind of change on the procedures. The change on the procedure are fully accepted in most of the countries because it introduces a quite add value to your process. Is as well very intensively used under PAT environment, PAT meaning Process Analytical Technology. I'm pretty sure, again, most of you are fully aware of those acronyms, but in case somebody is not familiar with them, I will give a very brief introduction of what PAT means or refers to. PAT refers to rapid and faster data collection. Basically, what we are doing is collecting data to have an understanding, processing the data to have a better, a more accurate process control of what is going on the production process. So what we are doing is taking more data to make a decision if we need to change things on the operation procedures. That is what PAT is about. It's an extra tool in the quality assurance that you have or introduce in your facilities. So PAT is always related to knowledge, okay? It's nothing more than knowledge that you are gaining, but that knowledge helps you to make better decisions on how to improve the productivity. Most of the time, the requirements to introduce technology as Raman Technology need to be based on a driving force that is going to be the requirements from the customers. Many times, the customers need to sit back and put the user requirements specifications together to justify, first of all, the investment, but then the reasons in front of the regulator why you want to introduce those changes in production. So many times, the customer needs to ask himself, what is the sensitivity that I need to have in the technology I want to use to have an answer into my production process? I need to have the sensitivity, selectivity, simplicity, stability, or specificity of the technology, for example, to replace an HPLC. Sometimes the answer is yes, sometimes the answer is, well, maybe it's not necessary to go to that level of sensitivity. It's not introducing any add value to my processes. So you need to understand those requirements as well to understand how far you need to go to introduce those changes. Always, there is as well, it's good to have the technology, it's good to change things, but as well there is a cost associated with those changes. So if you want to have a lot of information, there is plenty of technology today available on the market that can produce you tons of information. But as well, you want to have that access to that information at a reduced cost. So you need to balance all those elements before you start putting these together. So a clear understanding of the tool is a need to have a clear understanding what you want to achieve with all this extra knowledge that you can achieve in your production processes. Raman Technology is a technology based on light. Okay, we take a laser beam, as the one that you can see on my hand. It's just a polarized light, very precise wavelength that you need to use. The problem with the technology, there is always problems with all technologies, otherwise we will not have the variety of product that we have on the market. is that there is sensitive to fluorescent process. Fluorescence is natural process that there is in some molecules. So when you have Raman signature and there is fluorescence, sometimes the Raman signature is hiding behind the fluorescence. So you want to have a system that is as fluorescence-free as possible. So the fluorescence is, that is Raman spectrum of the same material. You can see that you have a deviation from the typical baseline of any spectrum. People like a spectrum where it's flat baseline with clear peaks. But when you have this fluorescent problem, basically what you have is deviation of that baseline that introduce an extra noise or background noise in your spectrum. So the selection of the wavelength of the laser that you are going to use, especially when you are going to do quantitative measurement, is very important. The intensity of the Raman peak, any peak that you have in here, is going to be directly proportional to the concentration that you have. So it's a quantitative technology, logically. But if you have some deviation introduced by noise, that may affect your measurements. So you need to be sure that you select the right wavelength where you are going to use Raman for quantitative measurement. Raman on P80, it is used today very extensively in chemical reaction monitoring. That is one of the areas where it is used the most, okay? This is the area where P80 and Raman are working the most together for quantitative analysis. But the technology is broadly used as well in just purely the identification of the chemicals that you have in there, just to know is the material that I'm looking for already in my reactor, for crystallization processes, distillation, drying and isolation, polymorphic screening, again, for example, for the patent users, as well as at aiding formulation. So is my granulation monitoring going okay? So may I have problems with the copolymerization agents, with the crystallinity agents that I'm using? All those areas are today areas where Raman spectroscopy is being used on the process control. But then the customer came to us as a manufacturer of a Raman technology saying, okay, I'm very interested in the information I can obtain from this technology, but now I need to understand better how I do it. Because I am doing today this by HPLC, by infrared technology, and then you are asking me to change this. I think there is a good value in terms of information I can obtain, what I need to do. But the first thing that the customer needs to refer is logically to the Pharmacopoeia and refers what I need to do quantitative Raman in my processing. Again, it's very well documented. So what you need to do is accept and identify the linearity, accuracy, the range, the specificity, and precision of the technology for the specific processing that you want to implement. And that needs to be logically documented and validated in the process before you can go into production. That is where most of the time the technology presents a limitation because the people have sometimes trouble to understand really what is the regulations regarding the introduction of the technology. It's good to have the information, but if I'm going to have a lot of trouble to understanding how to validate this, then maybe sometimes the people are a little bit scared. Nothing to be scared about. Is well understood for companies like B&W Tek. We know how to do this process. So remember, P80 is just not putting a probe inside the reactor. It's obtaining knowledge. It's about obtaining information and then implementing that information to make decisions on the productive process. Introducing Raman on P80 is based, first of all, and most important of all, on the previous knowledge that the company have about this production. So it's just a transfer of knowledge from the R&D environment that can be in-house R&D or can be R&D down, for example, in our university, to the production process. So when you put a probe in there on the reactor, you need to know what products you have. You need to have an understanding what is the signature of the different materials that you will confront when you need to analyze those data. Raman produce data, okay? Let's go always be clear with that. What we need to have is people that understand that data and make the decision based on those good data that can be obtained. Is a fast and reliable technology to maximize the yield of the operation that you have. Sometimes to heat a reactor, when you have a 1,000-liter reactor and you need to reflux the system for 2 hours, it's extremely expensive and time-consuming to have this ongoing in there. So if I can reduce the time by the reactor by half an hour, maybe it's something that is worth to consider because maybe I'm introducing a better yield, better crystallinity, better quality in my product. Maybe I'm reducing just simply cost because I need to heat less. So all those elements is what this technology in combination with PAT can introduce for our customers. But remember, PAT requires a strong knowledge. So the knowledge needs to come from somewhere. That knowledge can be from the in-house or can be from somewhere else. Let's go take some example. This is an example that comes from a company is working with a university. As I say, the knowledge in this case comes from the university that is going to be introduced later on the production process. So basic chemistry in here. So we have an nucleation here. So different processes. You can see the reaction time is quite different. Logically, those are not pharmaceutical environments, as you can see, this is clearly university people, but it doesn't change very much, okay? What is important is the information that we are going to obtain. And the information that we need to understand here is that we have a reactant that have a very distinctive fingerprint identification, that we have a second reaction that we can identify as well for some bands that we know what they are, as well as we have the identification bands relative to the product. Each product have a number of bands or peaks that will define that product. It's his signature, his fingerprint. So what you need to have is clear understanding of the different elements, because when I put a reactor, I'm going to have signal for everybody, okay? So that is what you see on PAT, is an accumulation of data over the time. But then when you start analyzing those data is when you start making sense of what you have in your reactor. So basically what we do in Raman is basically we analyze peaks or information regarding to the fingerprint of the molecules that we have. And then what we do on the PAT environment is plotting that information, giving a graphical visualization of what is going on the reactor. Here we can see, for example, that there is an induction time in the formation of the product, because if you remember the reaction, I think I did something, right? Yeah. The reaction you can see here is just basic an acidic nucleation of the ring in here. Basically what you have is induction time to generate the intermediates of the reaction. Then you have a sharp increase in the reaction, and then you can see that this process is slowly going up. So if you have a good understanding how to improve or reduce those times of nucleation or formation of the intermediate, you can introduce a beta factor in your yield of production, for example, okay? But when the molecules get a little bit more complicated, for example, when you start introducing enantioselective enantiomers in the production, maybe you have information of this is key to guarantee that you have the precise EE that is required in your processing, okay? And that is where you want to be sure that the information that you obtain and the samples that you have in your modeling is correct, because otherwise you can have incorrect information. Logically, the final product, they are normally mixed system where you have excess of reactants, where you have byproduct, where you have a lot of things. The technology is able to cope with that element. The only thing that is need to be teach the machine what to recognize the information, okay? So a clear example how this knowledge is transferred to the production facilities. This is an example from a company, Eli Lilly, that was presented last year in SciFax. Basically, they got the technology and they were replacing a gravimetric technology that was used to quantify some products and to transfer that to the production line. Basically, what the company was doing, taking a sample, send to the laboratory, do a gravimetry there, send the results back, the results were okay, I can go into production. There was few hours processes that was a waste of time, basically. The reactor needs to be ready, all the products need to be ready, everybody on the team need to be waiting for the sample to be sent to the laboratory do the result, and then you can go into production. Hours waste. While if you open your reactants, measure that what you have in there is what is supposed to be, and then just do the reaction. You save hours just by changing your procedure. But that is what this company did. And basically, they're using the B&W Tek product for Raman. It's a portable device. Just take the instrument, go there, and just do the measurement. Always keeping in mind that needs to be following perfectly the quantification protocols for validation in the pharmaceutical market. Again, remember, you need to study and demonstrated the linearity, accuracy, range, specificity, and precision of the technology. And then you can generate your modeling. Modeling means that you have a calibration curve that tells you the concentration that you have, okay? That is what it's all about at the very end. You need to have a number that is, yes, it's good to go into production, or it's not good enough to go into production. So generating the linearity with your modeling, generating the accuracy data, understanding the robustness, the problems that you can have when you do the measurement. Now it's mandatory, according to the FDA, to do risk analysis on every single step that you take in your production change. So it's a lot of thinking that you need to introduce, but it's relatively simple with this technology, okay? Evaluated the different elements. For example, in spectroscopy, it's very well known that you need to have, for example, lasers changes or probes changes. For example, in ERIR, if you change the probe, you need to change basically all your modeling. In Raman, it's not that sensitive. So Raman, you can change the probe without any problem, without affecting your models. So that is demonstrated, and it works. So you can obtain a lot of extra information with Raman into the PAT processes. You need to understand the protocols. You need to understand the needs of the technology. But there is companies like B&W Tek that we are here to help you. That is part of our business, okay? So quick conclusion. Raman is a very well deployed technology in the field, specifically in the pharmaceutical companies. Is one of the technology that is introducing the most today. It's a tool that requires validation, but is very valid for qualitative and quantitative, okay? Regulations are well understood. If anyone has any particular question on this area, I'm going to be here in a few days. I specialize in this area. And Raman P80 is one of the strongest sources of innovation today in the Raman as well as in the PAT process. Because working together, we can obtain certainly a much better future. And with this, I would like to thank you for your attention. If anyone has any specific or technical question, we're going to be in the hall 4J74, and I will be glad to have any question answered if you want to have any. Any questions? Okay, thank you.